Assalamualaikum dear students. Today we are starting our next topic, that is the biological nitrogen fixation. So what nitrogen fixation is? Nitrogen fixation is conversion of the molecular form of nitrogen, that is the atmospheric nitrogen, into ammonia. It is a reductive process, or you can say it is a reduction process when the atmospheric nitrogen or molecular nitrogen is converted to ammonia. This is a reduction reaction. And why it's called as biological nitrogen? Because it occurs by means of microorganisms which are prokaryotes. Only prokaryotes are able to fix nitrogen. Eukaryotes don't have any capability to fix nitrogen. So here is the reaction for the fixation of the nitrogen. It's the molecular nitrogen N2 which gets converted to ammonia. In presence of means the reaction requires 6 electrons plus 6 protons. Now only ammonia in this reaction is not synthesized. What happens? This biological reaction also generates at least 1 mole of hydrogen gas H2 in addition of 2 moles of NH3. Here 2 moles of NH3 are produced and it generates 1 mole of hydrogen gas also. But what happens? Here 6 electrons and 6 protons are required, but to produce 1 mole of 1 additional mole of this hydrogen gas, 2 additional electrons and protons are required. So the reaction becomes N2 gets converted to 2 moles of ammonia plus hydrogen gas, 1 mole of hydrogen gas, and this reaction requires 8 electrons plus 8 protons. Also it requires ATP. Fine. So this is the overall reaction of nitrogen fixation, where this molecular nitrogen gets converted to ammonia and hydrogen gas is evolved, and it requires 8 electrons plus 8 protons. So let me tell you here the enzyme responsible the enzyme which is required in this reaction is nitrogenase complex. It's a complex enzyme and it's called as nitrogenase complex, and it catalyzes the process of nitrogen fixation. You know, in every reaction is catalyzed by some enzyme. The enzyme catalyzes any reaction, and the enzyme required here is nitrogenase complex. Now what happens? This enzyme nitrogenase complex can exist in three different forms, and depending upon what? Depending upon the cofactor required. You already have studied in your chapter of enzymes that every enzyme requires some cofactor, metallic cofactor, and here this enzyme exists in three different forms depending upon the cofactor used. Sometimes it requires molybdenum, vanadium or iron as cofactors. So it can exist in three different forms depending upon the cofactor used. And the most common cofactor used by the nitrogenase complex is molybdenum. So please remember, these are the three cofactors upon which the forms of the nitrogenase complex depend. Now what happens? This nitrogenase complex, it's very sensitive to oxygen, and what happens if it's exposed, if nitrogenase complex is exposed to oxygen, it gets irreversibly inactivated. It gets inactivated by oxygen and does not revert back to its original form. So this complex, this enzyme is very sensitive to oxygen. So what we conclude from this? We conclude that the nitrogen fixation should occur in anaerobic conditions. Anaerobic conditions, oxygen should not be present. Why? Because the oxygen inactivates it irreversibly. So here, let me tell you, it occurs under anaerobic conditions. Fine. So what happens? Facultative anaerobes. Facultative anaerobes are those organisms which can synthesize ATP in presence of oxygen if oxygen is available, but can survive without the oxygen. Means they are facultative anaerobes depending upon the conditions. Sometimes they behave as aerobes, like they can synthesize ATP in presence of oxygen and but can survive in absence of oxygen. So these are called as facultative anaerobes. What they do, they synthesize, they fix this nitrogen in anaerobic conditions. When they have to fix molecular nitrogen, they create anaerobic conditions. So these are the facultative anaerobes. Now those aerobic prokaryotes, how do they fix nitrogen? Oxygen is present in them, oxygen is required for their respiration. How do they fix the nitrogen then? How do they fix? They have specialized cells in them, and those are called as heterocysts. Please remember, in aerobic prokaryotes, fine, which require oxygen for their process of respiration, how do they fix the nitrogen then? They fix the nitrogen in specialized cells called the heterocysts. You must have already heard somewhere about heterocysts. What are heterocysts? These are thick-walled cells which lack photosystem second. These are the thick-walled cells which lack photosystem second. They don't have any photosystem second. You already know the splitting of the water takes place in the photosystem second, when water is separated to oxygen. So they lack the photosystem second. As a result, oxygen इनमें नहीं होता है. They don't have any oxygen, they don't produce any oxygen, so they don't generate any oxygen. As a result, nitrogen fixation occurs in these cells under the anaerobic conditions. So please remember, like I will give you examples of these are aerobic prokaryotes which fix into in specialized, for example, cyanobacteria or blue green algae, you can say. Fine, I hope till now I am clear. So moving forward, nitrogen fixing prokaryotes are of two types. Nitrogen fixing prokaryotes are of two types. Number one is free living prokaryotes. They don't have any association with any plant. They are free living. And what they do, they fix atmospheric nitrogen into ammonia and then release into soil and all that. Later plants assimilate that out of the soil. So, number second are symbiotic nitrogen fixing prokaryotes. Symbiotic nitrogen fixing prokaryotes are those which establish a relationship with the plant, and that relation is symbiosis, means the organism here, the prokaryote is also benefited and plant is also benefited. The plant gets benefited in the manner that it gets the nitrogen, fixed nitrogen, and the bacteria or the prokaryotes gets benefited in the manner that it gets the food and shelter from the plant. So about free living prokaryotes, let me tell you, anaerobic aerobic free nitrogen fixing bacteria, aerobic, those who are aerobic and fix nitrogen, for example इसमें जो example है azospirillum and azotobacter. Please remember, these may be asked in your exams. Azospirillum as and azotobacter, they are aerobic nitrogen fixing bacteria. And number second are anaerobic nitrogen fixing bacteria. For example, we have rhodospirillum and clostridium. These are anaerobic nitrogen fixing bacteria. So how nitrogen fixing takes place in aerobic? They fix nitrogen in specialized cells called the heterocysts. Fine. And in symbiotic, what they do, they establish a relationship with their plant. They form or not form the those are called as nodules. So tomorrow we'll be studying the symbiotic nitrogen fixing prokaryotes. Fine. So I hope I'm clear till now. If there are any queries, please let me know. Thank you so much. Stay blessed and stay safe.